All right, welcome to part 5, modeling addiction. How do we do this, right? How do we study addiction in the lab in an animal model? Well, I mean, you can't give, rats some packets of cocaine and a nice little Coke mirror and expect it to go to town. We have to do things a little bit differently than we would do with the human population. So let's talk about reinforcement. Most drugs are positive reinforcers rather, which means we're adding something to the rat experience, and consuming the drug strengthens the preceding behavior. Drug reward is a positive experience that's associated with the drug, right? So how do we know if a drug is reinforcing? Right, the rat can't exactly tell us. We can't ask the rat, hey, did you like that bump of cocaine? We have other ways of finding this out. So a lot of what we know relies on self-administration models. You can see here from this figure that basically the way this works is that a rat is surgically implanted with a catheter, which will deliver a drug directly into its system. And then we can sort of hook this up to a computer that is going to regulate the dispensing of this drug. So when the rat does something, typically something like a lever press, it'll be recognized by the computer as an input, right? Okay, a rat has pressed lever, which will advance this syringe pump and deliver a set amount of the drug to our cell. So as I mentioned, rats are catheterized, and that is they're implanted with a catheter and trained to perform a behavior to receive a dose of a drug. Very typical is a lever press. Typically a fixed ratio reinforcement schedule produces an inverted U dose response function. So what does all that mean? A fixed ratio means that a reinforcer delivered after a fixed number of responses. So rat pushes lever X number of times and receives a reward, right? The infusion of this drug. So this lets us measure how hard is a rat going to work to receive that drug. Low doses don't prompt much responding because it's not very reinforcing, right? So if you're giving a rat just a tiny amount of drug, let's say cocaine, give a rat a tiny amount of cocaine, it's not going to work very hard for that. That's not very reinforcing. Moderate doses prompt very, very high levels of responding, right? Reinforcing, but they're not seated quickly. It's enough of that drug to say, oh, hey, that was good. I want more of that. I'm going to push a lot to get that. High doses prompt very low levels of responding, right? This is for a number of reasons, right? One, rats are surprisingly good at deciding how much of a drug they want to have. They don't really want to overdo it, so they might become sated, right? They don't have to push very many times to receive enough of that drug. They've had enough, they're good. Also, an aversive reaction could be produced, right? Too much of a drug can be a bad thing. So the rat might give itself too much and say, oh, wow, that was bad. I don't want any more of that. And behavioral impairment is also an issue. Many drugs will produce locomotor problems at high doses, prompting a low level of responding. That's why we call it an inverted U function, right? Low at the low end, high in the middle, low at the high end. So here are some inverted U functions for two different drugs, cocaine and heroin, at different doses. Right, so different classes of drug will prompt different shapes of curves, but generally speaking, they apply the same sort of shape, right? At a low dose, you'll see a low level of responding. At a moderate dose, you see a high level of responding. And at a high dose, you see a low level of responding, right? For the reasons we talked about on the previous slide. Okay, so that tells us basically whether or not a drug is reinforcing, but what if we want to see how reinforcing is it? How hard is our subject willing to work to receive that drug? So that's what breakpoint studies are designed for. A breakpoint involves training under a progressive ratio schedule, which is what it sounds like. Progressive ratio means the number of responses required increase with each reinforcer obtained. So it might take 1 press for the first, then 2 for the second, then 4 for the third, and then 8, 16, and so on. Right? So the number of presses required to earn a reinforcer gets higher every time a reinforcer is earned. The breakpoint is the highest number of responses the subject will perform to receive that reinforcement. And this will escalate as a function of dose per injection, right? So the higher a dose that's being received, the more reinforcing, in theory, and the harder an animal is going to be willing to work for it. So if something's not very reinforcing, a small dose, let's say, the breakpoint's going to be pretty low. Rat might press a couple of times and then say, hey, it's not worth it. I don't really like it that much. As that dose increases, or as the drug that we use becomes more reinforcing, the animal is going to be willing to work harder and harder and harder, pressing hundreds or perhaps even thousands of times to receive that reinforcer. Basically, what we infer from this is that the harder an animal is willing to work, the more reinforcing the drug must be. Another way to ask the question, does the animal like this substance, is what we call conditioned place preference. This is a kind of a clever task. It's very simple. You have an apparatus with two chambers. These chambers look different. You might have horizontal versus vertical lines and a different textured floor. And basically what you do is you give the animal an injection of your drug of interest and place it in one compartment. And then the next day you bring them back, give them an injection of a control substance, saline for example, and then slap them in this compartment. Let them sit there, chill, and experience the environment. Do this a few times, alternating days, and then on your test day, you place the animal in the center compartment and remove guillotine doors, right? So normally these compartments are separate from one another for the test. You open it up this little hallway, and the rats can choose which compartment they would like to sit in. The thought is that the rat will come to associate the context with its subjective experience, right? And if the rat liked the experience of being on that drug, it will favor that compartment. So let's say we give a rat cocaine and put it in this side, and then we give it saline and put it in this side. Then on the test day, we let it choose which compartment it goes into. If the rat enjoyed its experience of being on cocaine, which most rats do, it will slide over to this compartment and hang out there for most of the test. That shows us that the rat found that drug reinforced. So there are many interesting questions to be asked with animal models of addiction. How do animals learn about cues that predict things? And how do animals substitute one drug for another? What is their internal state like? But one of the most common questions asked is about relapse. So we have models for relapse in self-administration. So let's talk about that. Basically, how this works is subjects are trained to perform behavior in order to receive a drug. After that, the animals undergo a period of forced abstinence. After that, we have the introduction of some stimulus that provokes renewed responding. We call this phenomenon reinstatement. It's analogous to the human phenomenon of relapse, right? Falling off the wagon after a period of abstinence, going back to drug use after being drug-free for a while. There's a couple of ways that this can be accomplished. Classically, these are giving a small drug dose, which is known as drug priming. Stress, so causing the animal to be stressed out, will cause it to renew its responding, looking for that drug again. And then exposure to cues that were previously paired with drug delivery. So if you turn a light on in the environment whenever they receive a drug or play a sound, exposing the animal to that, again, will cause reinstatement. So here's what that would look like in terms of data. So here we have graphed the time period, self-administration days 1 through 10. So this is the time when the rat is able to press a lever to administer a drug to itself. And then here we have extinction. Extinction means that the lever that they've pressed to receive the drug is present, but pressing the lever does nothing. So here's what we see. And in red, we have our drug infusion lever, which is the lever they press to receive the drug. And in blue, we have a control lever. It's basically a lever that's not hooked up to anything. It has never provided a reward. It will never provide a reward. So here's what we see. Animals learn pretty quickly to press that drug infusion lever to give themselves doses of that drug. And after they've acquired the behavior, they press pretty steadily. However, we put them into extinction where they can no longer receive the drug for pressing, and their drug-seeking behavior declines, right? If pressing the lever doesn't get them any drug, they stop trying to do that, right? And after a while, their responding is very, very low. They're no longer pressing that lever. However, here, we can cause reinstatement, or a model of relapse, right? We can show them a cue. If, for example, a light went on every time they pressed the lever and received drug, we show them that cue again. If we could also subject them to stress, maybe food restriction or expose them to some foot shock, something like that, to stress them out a little bit. That will cause them to renew their responding. Or drug priming. Perhaps in their home cage, we give them a little dose of the drug they've been working for. We put them back in the environment, and suddenly they're working hard to try to get that drug. So it doesn't take much imagination to imagine why we care about this, right? These are a model for human relapse, right? We know that When people are drug-free for a while, there are a number of things that can cause them to start seeking drugs again. One is exposure to cues that have been associated with that drug, right? Seeing people they used to use with, or drug paraphernalia that is associated with using that drug, or hanging around the old neighborhood, right? These things are cues that could remind them of drug use. This is just basic classical conditioning, right? Things that are become actually through learning mechanisms associated with drug use, right? Paraphernalia, for example, that you use to take drugs becomes associated with that drug. Seeing that cue will elicit the conditioned response of drug craving. Likewise, stress, right? This can lead people and animals to start seeking using drugs once again. And drug priming, a small exposure to that drug can lead to drug seeking again. One of the reasons why people are abstinent, so for example, people who are former alcoholics and recovering usually don't drink even casually, right? Because even a small dose of something that they have been avoiding can cause them to crave it once again. So we have talked about tolerance already, but I'm going to just run through it again very, very quickly. So tolerance is basically the idea that with regular use of the same drug, the effect will diminish. Larger doses then must be taken to achieve the same effect, and there are three primary types. First up is drug disposition or metabolic tolerance. This is when repeated use of a drug results in less of that drug being available at the target tissue, right? The drug increases its own rate of metabolism and becomes more efficient at that metabolic process. So basically, when you take a drug, less of it gets through your first pass metabolism, and because less of that drug is available, it has diminished effects. Pretty simple, right? Taking drug increases metabolism for that drug. Then in the future, less drug gets through, diminished effect. Pharmacodynamic tolerance is a bit more complicated. Repeated use of a drug changes neuron function, things like receptor density and cascades that happen within the cell after that signaling. This can look a number of different ways, right? We could have a chronic reduced receptor activation following taking an antagonist. This will result in receptor upregulation, right? As a compensatory mechanism. If you're taking a drug that reduces receptor activation, we might see more receptors present. The opposite could be true for an agonist, right? If you're taking a drug that increases receptor activation, we might see downregulation of those receptors. You see this with stuff like opioid tolerance. Synaptic changes can also be referred to as functional tolerance. So basically, pharmacodynamic tolerance is a compensatory shift, right? You change the way that your neurochemistry works by elevating or decreasing the level of some neurotransmitter or some receptor or something, and your brain does its best to compensate for that by turning up something else. This leads to a lot of those compensatory mechanisms that we talked about. Oh, that's not good. So behavioral tolerance is repeated use of a drug, reducing its effects in the environment where the drug is administered, but not in other environments. So this is just basically classical conditioning at play here once again, right? Cues in the environment or paraphernalia being used serve as sort of conditioned cues for what's to come, the effects of that drug. Our bodies use those cues to start compensatory processes, right? Things that might counteract the effects of the drug dose that's to come. That's a form of tolerance. And one of the reasons why we see drug addicts overdosing on a standard dose in a new environment is because those compensatory mechanisms don't take place in a novel environment. So a standard dose could prove to be an overdose without this tolerance in play. Okay, and here's a graph taken from a psychopharm textbook showing the types of tolerance exhibited by certain drugs. So not every drug is going to result in the development of every sort of tolerance. Some produce all forms of tolerance, and some produce far fewer forms of tolerance. So it varies very, very much depending on the mechanisms of that drug. Okay, that's it for this unit. Next time we're going to be discussing a couple of drugs of interest.